All right. Hi, everybody. This is Ryan Kuhl. I'm here with Brittany Anderson, April Bean, Jessica Evans, Sam Marshall, and Megan Tursty. We're going to be talking about the cell biology of alpha-1 antitrypsin deficiency today. Next. All right, a little background on the alpha-1 antitrypsin and its deficiency. There is a genetic basis to this, which is found on the SERPINA1 gene. Now, SERPINA1 can be found on the long arm of chromosome 14, as you can see there on the graphic. There's multiple mutations that can affect SERPINA1 and therefore affect the disease. So depending on the allele that is inherited, you can have a variation in symptoms and severity. Next. A little more background here on alpha-1 antitrypsin. So normally it acts as a protease inhibitor. And so it inhibits those enzymes that break down protein, elastin, and collagen, which is a good thing because all those enzymes can be helpful in times of infection or tissue repair. Unregulated activity can lead to tissue damage and inflammation. And so alpha-1 antitrypsin has an important role in controlling that. So more specifically, it regulates the action of neutrophil elastase. Oh, can you go back? It regulates the action of neutrophil elastase.
Yeah.
And it is produced in hepatocytes, which are liver cells, and so it travels through the bloodstream to the lungs where it acts on neutrophil elastase. Next. So a little more background on the disease. Mutations in SERPINA1 lead to a defective form of AAT, so it changes that conformational shape of the protein. And so oftentimes this defective AAT polymerizes within the hepatocytes. So you can see on the lower half of that graphic there that those red clusters are little granules of polymerized AAT that are stuck in the endoplasmic reticulum of those hepatocytes. And so this can lead to endoplasmic reticulum stress, which can ultimately lead to apoptosis of those cells. And so it's not uncommon to see liver damage or cirrhosis of the liver there with this deficiency. Also, the second part of this is lung damage. And so without having AAT in systemic circulation, neutrophil elastase in the lungs goes unregulated. And so it starts to break down the elastin in the lungs, leading to pulmonary issues. Next. So a little bit about the prevalence of AATD. It's not a very well-studied topic. However, we do know that it is most prevalent in European individuals and much less common in those of Asian, African, or Hispanic descent. And a study found that a small island off of Portugal called Madeira has the highest prevalence of this, with nearly 41 people per 1,000 having a genotype for AATD. Next.
Okay, so the symptoms can be divided in two categories. Symptoms from lung damage include chronic cough, fatigue, ongoing lung infections, shortness of breath, and breathing problems. Symptoms from liver damage can include jaundice, swelling in the legs and abdomen, weight loss, lack of appetite, nausea, pale stools, problem nursing as an infant, and cirrhosis. Some patients also reported depression, anxiety, stress, and pain. Your symptoms will differ depending on what type of allele you have and your lifestyle factors and environment. Next. Now, this is just like a representation of what kind of leads to the symptoms, and you're healthy. Your alpha-1 protects your lungs, and in the healthy liver, it releases alpha-1 into the blood. And in the unhealthy side, the antitrypsin deficiency leads to lung tissue damage, and the buildup in the liver leads to cirrhosis. Next.
So there are, as Jessica mentioned, there are different types of alleles that can be inherited with this disease. And there are three main types, PIM, PIZ, and PIS. PI just stands for protease inhibitor. As Ryan described, it means that these alleles produce proteins that break down or inhibit the breakdown of peptides and other proteins. The most common allele is PIM allele, and that is the most common in all humans, and it produces the normal form of protein. This protein has a few key components, mainly that it has 3 beta-plated sheets, one right here, here, and here, which is kind of hard to differentiate, as well as 9 alpha helices, shown in gray, and then also it has asparagine-linked carbohydrate side chains, shown in yellow, at positions 46, 83, and 247, as well as an RCL, which stands for reactive center loop, and that is where it interacts with neutrophil elastase to inhibit its activity in the lungs. And then there's also the PIZ and S allele, which are the most common mutated forms of the allele, and Z being the more common of the two. The Z allele is produced due to a mutation at the 342 residue site, and it's a switch from glutamine to glycine at this green point right there. And this causes a change in shape of the protein that causes slower folding and higher sensitivity to temperature. And due to this, it also causes changes in location of the alpha helices, which can be seen especially between these two. And also it breaks up the RCL from the 5th strand of the beta sheet A. And due to its opening, beta sheet A can then bind to an RCL group of another mutated protein within the liver and cause polymerization to occur. And these long chains are called long loop polymers, and these can build up in the liver to cause stress in the ER, as well as PIS also produces a different mutation at the position 264, and this mutation causes an unstable protein that gets quickly degraded outside of the liver, so it's able to escape the liver, unlike the Z allele. And then additionally, the null allele does not produce any AAT at all, and so therefore you will have mostly effects in the lungs rather than the liver. And there are over 30 other variants, but they're very rare and only make up 5% of cases. So we're going to focus only on these 3 types of alleles.
So the 2 most common mutations in SERPINA1 are the protease inhibitors Z and S. The Z allele leads to the misfolding and polymerization of AAT. Polymerization of the protein inside the ER causes there to be an accumulation of proteins in the ER lumen of the liver cells. This accumulation causes ER stress, ER overload response, which involves calcium signaling and pro-inflammatory response, and autophagy, so apoptosis. These responses begin to cause damage to the liver. The S allele, which destabilizes the AAT, misfolds the protein, retaining it in the liver cell ER lumen, causing ER stress, which triggers ER-associated protein degradation, or ERAD, and the unfolded protein response, UPR. The S allele does not form polymers unless the Z allele is present. So the image to the right shows ER stress and the unfolded protein response. In the far right of this image, it shows a normal cell process involving AAT, where the properly folded proteins, indicated by the green arrows, are processed in the Golgi apparatus and then translocated to where they're needed in the body. Misfolded proteins like the S allele mutation, indicated by the red arrows, are retained in the ER lumen and degraded through the ERAD process, where the proteins in the ER are retranslocated to the cytosol for proteasomal degradation. Mutations like the SERPINA1Z mutation causes an accumulation of misfolded polymerization proteins in the ER lumen, causing ER stress, indicated by the blue arrows. In response to ER stress, the cell activates the unfolded protein response, in which accumulated misfolded proteins are sensed by incital requiring enzyme 1, represented as IRE1, activating factor 6, represented as ATF6, and protein kinase are like endoplasmic reticulum kinase, represented as PERK proteins. IRE1 protein dimerizes, which removes a small intron of the transcription factor X box binding protein 1, represented as XBP1U, that is then converted to XBP1S, which acts as a transcriptional activator. ATF6 is cleaved and activated in the Golgi apparatus to yield a transcription factor, ATFF6C, that migrates to the nucleus where it is activated, which activates the transcription of UPR target genes. PERK dimerizes and phosphorylates the eukaryotic translation initiation 2 alpha, represented as ELF2 alpha, which attenuates most translation but stimulates translation of the transcription factor ATF4, which in turn activates genes to protect cells against ER stress. The UPR signaling consists of 4 mechanisms. The first is decreased translation to prevent further misfolding protein accumulation. The second is induction of ER chaperones to increase folding capacity. The third is induction of ERAD genes to increase degradation of misfolded proteins. And the last one is the induction of apoptosis to remove stressed cells. Next. Although most of the misfolded proteins are eliminated, a portion of the correctly folded proteins are secreted into the circulatory system and travel to the lungs. The consequence of this is that there are lower amounts of AAT circulating to the lungs, meaning their intended function of inhibiting neutrophil elastase is not accomplished. Next.
All right, so AATD is actually a pretty unique disease among other protein misfolding diseases because of its mechanisms that can cause its severe symptoms. So seen in this graphic here, the mutation of AAT incorporates both a toxic gain of function within the liver as well as a loss of function within the lung, and these can result in liver cirrhosis due to the PiZ mutation and emphysema due to the lack of AAT in the blood. So we will break down each of these mechanisms to kind of explain why these diseases occur and how they occur for patients with AAT. So for the toxic gain of function found in the liver, the misfolded AAT proteins involved are actually transcribed by the inherited PiZ allele. So within the liver, the misfolded proteins polymerize and are retained within the ER of the hepatocytes. The aggregation of all these misfolded AAT proteins disrupts the function of the ER and induces ER stress. Now, this stress activates a signaling pathway that kind of helps the cell to adapt and respond to this aggregation of misfolded proteins. However, with prolonged ER stress, apoptosis signaling is activated. So the accumulation of AAT proteins actually leads to the ER stress-induced apoptosis of the hepatocytes. Next. And this apoptosis from the toxic gain of function mechanism can result in chronic liver disease, liver scarring, cirrhosis, and in some cases, hepatocellular carcinoma. And as stated previously, the toxic gain of function in the liver only occurs from the PiZ allele. So the PiS allele is different from this PiZ allele because this protein is actually able to be secreted from the ER. However, it is still a misfolded protein and thus is degraded from the ERAD system that was explained earlier, leading to both of the alleles having a lack of AAT in the bloodstream. And because of this lack of AAT in the blood, AAT, the protease inhibitor, is unable to inhibit neutrophil elastase seen in the graphic. So the unregulated protease activity of this enzyme can actually damage the lining, connective tissue, and alveoli of the lung through the uninhibited elastin breakdown. Next. Now, this damage from the loss of function mechanism can result in early development of pulmonary emphysema from damaged alveoli and COPD, which causes obstructed airflow. And here is just an overview of the mechanisms leading to the symptoms of AATD. So the AATS proteins are secreted from the ER and degraded through the ERAD system. And the AATZ polymers lead to ER aggregation and stress, resulting in liver disease, cirrhosis, and carcinoma. And both mutant S and Z proteins lead to decreased levels of AAT in the blood. And AAT protects the lung from inflammatory mediators such as neutrophil elastase, but with less AAT, there is uninhibited protease activity, leading to increased elastin breakdown, resulting in emphysema and COPD.
Cool.
So when it comes to the variants in symptoms, it really depends on the type of allele being expressed, or the genotype. So the genotype is also directly related to how much AAT an individual is missing, and it kind of helps vary the age of onset and severity of the AATD. So in this graph, there is a range of AAT in the bloodstream for each genotype, as well as the risk of emphysema and the risk of liver disease. As you can see, for the wild type MM alleles, there is no risk. With one Z allele, there's increased risk of lung and liver damage. And with two S alleles, there's only risk for lung damage. And as shown previously and in this graph, only the Z allele can lead to both liver disease and emphysema, making those who have inherited Z alleles from both parents at very high risk, and they may require a liver or a lung transplant in the late stages of AATD. And the very, very rare null alleles, also seen here as well, result in the production of no AAT at all, only leading to a very high risk of emphysema. Now, some individuals with severe deficiency of AAT never develop emphysema and can have a normal life, especially if they never smoke. So life factors like drinking and smoking can increase the severity of your symptoms. And those with AATD who smoke or are exposed to a lot of pollution are at increased risk of developing lung disease. However, for those who don't smoke, especially with a less severe genotype, they can have a more normal lifespan. And in this graph shown here, it shows that smokers and non-smokers with a severe ZZ genotype and their probability of survival. And the probability of survival for those that smoke is dramatically decreased over time compared to those that never smoke.
So the inheritance pattern of AATD is considered autosomal codominant. It is considered a non-Mendelian genetics because it doesn't follow normal autosomal dominant or autosomal recessive patterns. In codominance, both alleles express protein. So for example, in our diagram on the right, we have a cow with a red coat color and a cow with a white coat color. Each of these are going to inherit, this cow is going to inherit both a red allele and a white allele from their parents. This cow is also going to only produce red alleles from this allele, red proteins from this allele, and white proteins from this allele, leading to a mixed coat color of both expressed red and white proteins. Therefore, with our disease, depending on what allele is given to the offspring, then both of those alleles will be expressed. For example, in this diagram, as Brittany previously talked about, individuals with homozygous M alleles will produce both normal proteins, so they will not experience any symptoms of AATD. However, those with heterozygous genotypes will produce both normal and mutated forms of the AAT protein. However, these individuals are considered to be silent carriers because they tend not to show any symptoms of the disease, and that's because the normal allele is able to carry out the functions even though it's only working at half of what it could be. Finally, patients with homozygous Z alleles or S alleles are producing both mutated proteins from both alleles and therefore exhibit higher degrees of symptoms compared to heterozygous or homozygous for M. The inheritability pattern can be examined through this chart. If two individuals that are heterozygous for the M and Z alleles want to have or produce offspring, they have a 25% chance of producing offspring with completely normal genotype, 50% chance of passing the heterozygous genotype down, and 25% chance of giving their offspring the AATD deficiency.
Okay, so in summary, AATD occurs due to mutations in the SERPINA1 gene at chromosome 14. This leads to the secretion of mutant AAT proteins, and this is inherited through an autosomal codominant pattern. As shown in this picture right here, as Brittany and Megan both stated before, this leads to, if both parents are carriers, it leads to 25% of an offspring being AATD or having AATD. This disease most commonly affects individuals of European descent, about 1 in 15 to 3,500. And PIZ, the most common mutated allele, causes the most severe form of AATD. Next. So the accumulation of misfolded AAT proteins in the endoplasmic reticulum of hepatocytes can lead to liver disease. And due to this inability of mutated AAT exit the liver, it cannot reach the blood, and neutrophil elastase activity is unopposed. Therefore, and as seen in this picture to the right, elastin in the lungs is actively broken down, and this leads to lung damage and disease because after expansion and recoil, the lungs are not able, or expansion and contraction, the lungs are not able to recoil. AATD is the major known genetic risk factor for lung disease and a prominent genetic risk factor for liver disease. It can be diagnosed through blood and genetic testing. And next. This is a little overview of the pathology. So you get a SERPINA1 mutation. This leads to defective AAT, which builds up in the ER of hepatocytes. Over time, this induces ER stress and liver damage. Because of that, it cannot enter systemic circulation. Therefore, it's absent in the lungs, and unregulated protease activity in lungs, such as neutrophil elastase, and this leads to lung inflammation and disease. Approximately 10% of infected infants and 15% of adults with AATD develop liver disease and/or cirrhosis. This is the most common genetic diagnosis in children, and several symptoms are also arise because of this are associated with liver and lung disease. Another rare symptom is paniculitis, which is a rare skin disorder. Two pictures shown here. At the top right is a liver biopsy, and the pink globules, you see, as Ryan mentioned earlier, are a characteristic of this disease. And the other picture shows a patient with emphysema caused by Alpha-1 antitrypsin deficiency. Lifestyle factors such as alcohol and smoking can cause variety in these symptoms.